Good morning, everybody. Monica and myself, Ximena Londoño, we are going to present the result of the research doing by Luciana Díaz and her team, professor of the Universidad Javeriana in Bogota. Luciana, at the last moment, she wasn't able to assist to this Congress, and she asked me to present her talk. And Monica also agreed to help me.
Okay, so just to make things clear, I am not the author, I am just lecturer. So bear with me if I make any mistakes because this is totally out of my usual topic. Yes. So the title of this research is Guadua in Neotropical Lowlands, relationship between total phenolic compounds, microbiome, mycorrhization, and soil nutritional content in different species of the genus Guadua Poaceae in Colombia. So, this Guadua is a neotropical lowland genus comprising 35 species distributed in Mexico to Argentina and Uruguay. This research focuses on 8 specific species: Guadua angustifolia, Guadua aculeata, superba, amplexifolia, Weber-Baueri, uncinata, incana, and Venezuela, most of which are native to Colombia. The phytobiome can be understood as a complex network of interactions among plants, their environment, and the diverse communities of organisms, both macro and micro, inhabiting or associated with the plant. These communities profoundly influence the health and productivity of the plant within a specific ecological context. In particular, the assemblage of microorganisms associated with plants has generated significant attention. Regardless of the organ under study, it harbors a microbial composition shaped by numerous biotic and abiotic factors. As illustrated in the image, the soil associated with plants holds particular interest. It serves as a reservoir from which plants can selectively recruit microorganisms, acting as a repository of functional species. However, this recruitment process is also influenced by interactions among microorganisms themselves and their unique abilities, such as displacement and colonization of plants. This is especially relevant for microorganisms with endophytic lifestyles, such as certain bacteria and mycorrhizal fungi. Once the microbiome is recruited and established within the plant, which is shown in orange, it bestows benefits upon the plant. This one is shown in green, through various mechanisms enhancing its overall fitness. These mechanisms include the production of phytohormones, nutrition uptake, e.g., phosphorus, zinc, iron, stress tolerance, and resistance to pathogens. In this context, as shown in the image on the left, the interplay between the plant and its physical environment, particularly with the microbes residing within or associated with its surface, induces alterations to the phytochemical composition of secondary metabolites in plants. As previously mentioned, these metabolites fulfill various roles, including defense against pathogens, pests, herbivores, response to environmental stress, and mediation of interactions between organisms. As illustrated here, metabolites have undergone extensive study, particularly in Asia, across numerous bamboo species, showcasing the antioxidant capacity of phenolic compounds in various plant organs, particularly in leaves, as evidenced by the graph on the right. Among the myriad of environmental factors influencing plant secondary metabolites, altitude has been identified as a significant contributor to variation in flavonoid profiles among plant populations of the same species. While not clear separation was observed between the three altitudinal groups, low, middle, and high. As shown in the first image on the left, notable differences emerged when comparing populations from high and low altitudes, as illustrated in the lower portion. The comparison involved accessing the phytochemical profile of leaf extracts from 12 bamboo species, including 6 Guadua species, distributed across different elevation. The OPLS-DA score plot for the selected 12 species highlight the significant impact of evaluation on the metabolic diversity of bamboo populations, emphasizing the environmental influence on the metabolic profiles. Especially, the observation pattern suggests an increase in the flavonoid profile at higher altitudes, while lower altitudes are associated with elevated cinnamic acid-derived profiles. As shown in the image on the left, showcasing metabolites most strongly correlated with altitude. This phenomenon may be attributed to the heightened intensity and prolonged exposure to UV radiation at higher elevation, promoting the expression of secondary metabolites like flavonoids as a defense mechanism against adverse environmental conditions. However, further research is warranted to exclucidate the precise nature of this correlation, considering potential influences from other environmental factors such as temperature or soil properties. Among the studied species, Guadua angustifolia exhibited the widest altitudinal range, being sampled in 3 out of the 4 departments studied: Nariño, Quindío, and Cundinamarca. This species displayed a positive correlation between flavonoids and higher altitude, whereas the other Guadua species demonstrated a positive correlation between cinnamic acid-derived profiles and low altitudes. The box plot illustrates the behavior of select representative compounds from the group of flavonoids and phenolic acids across 2 altitudinal ranges evaluated for the studied bamboos. Hence, recognizing the significant impact of the microbiome on the metabolic profile of bamboo species under study, Lucía and her team conducted initial assignment of the bacterial community and arbuscular mycorrhiza forming fungi. Using high throughput sequencing technique, source samples were obtained for Guadua amplexifolia, angustifolia, incana, and uncinata. Their analysis revealed that while these species may share similarities in terms of richness and taxonomic composition, notable differences exist. For instance, archaeospores were absent in Guadua incana, whereas Redekera was present in Guadua amplexifolia. Moreover, the relative abundance of each taxonomic group varied considerably among species, suggesting a selective preference for specific groups depending on the species. In general, Glomeromycota fungi were dominated by the genera Glomus and Paraglomus, while proteobacteria and firmicutes were prominent among bacteria. These variations, as shown in the stark bar diagrams, were more pronounced among genera of Glomeromycota fungi compared to bacteria genera. Given the medical significance and phytotherapeutic potential of flavonoids, the study also compared the abundance of flavonoid profiles across populations of selected bamboo species by constructing a molecular network centered on their flavonoid metabolome. The color convention in the network highlights each species of Guadua, with Guadua angustifolia notably standing out, as indicated in the red box, for the abundant flavonoid profile. Particularly rich C glycoside flavonoids associated with asparagine, and this species exhibited distinct compounds such as evotexin and rheophylline, as illustrated in the figure. These findings not only underscore the vast metabolic diversity of Guadua, but also underscore the potential for exploring alternative uses of these species based on their biological properties. Further investigation into these components of the phytobiome that may modulate the diversity, especially in the case of Guadua angustifolia, holds promise for the future research endeavors. In their 2023 study entitled Metabolomic Profiling of Guadua species in the and its correlation with antioxidant and cytotoxic activities, Chitva and colleagues determined that Guadua angustifolia possesses the most intriguing flavonoid profile. This characteristic could be further enhanced through extraction optimization, as evaluated by both DPPH and ABTS techniques for both non-optimized and optimized extracts, as shown in the figure on the left. These findings pave the way for its potential utilization across various industries. This was further supported by chromatographic analysis and the exact extracts revealing that optimization notably increased the abundance of phenolic compounds in the profile of Guadua angustifolia. In order to elucidate the symbiotic association between Guadua angustifolia and its arbuscular mycorrhiza forming fungi, morphological analysis of their structure was conducted to determine the percentage of fruit colonization. However, to delve deeper and discrimination between the types of colonization that the fungus can exhibit with the root cortex of the plant was deemed necessary. Preliminary evidence suggested that in Guadua angustifolia colonization by Glomeromycota falls with the intermediate aurum pari category. Thus, growth pattern of each type were identified in the roots of sampled population. In the first type, aurum colonization picture above left, the fungus grows intercellularly, forming intercellular arbuscules that serve as the symbiotic exchange matrix. The second type, Paris type colonization, pictured in the center, involves intercellular fungal growth, forming arbuscules which can dedifferentiate and fulfill the symbiotic function. The third type, illustrated below, encompasses both aforementioned morphologies along the continuum that can arise from them. Confirmation of intermediate colonization by Glomeromycota in the roots of Guadua angustifolia revealed variations ranging from 26 to 96% across the population, signifying significant differences and indicating a notable degree of affinity between Guadua and arbuscular mycorrhizal fungi. Additionally, it was observed that in most cases the proportion of coils in Paris exceeded that of arbuscules in Arum, and an expected phenomenon under natural field conditions. This observation holds potential significance in relation to the content of phenolic compounds or other metabolites of interest, which may play a role in significant pathways favoring symbiosis or responding to stress induced by colonization. Various studies and approaches have acknowledged the presence of a complex network of factors converging with the phytobiome, influencing the relationship between plants and their environment. Armed with this knowledge, Luciana's group embarked on the initial inquiry posed by the title exploring the interplay between phenolic compounds, microorganisms, and soil properties, focusing on Guadua angustifolia. They endeavored to synthesize this information into a comprehensive evaluation matrix, elucidating the statistical relationships among phenolic compound content, mycorrhization metrics, key microorganisms associated with the species, and soil and climate properties, totaling 49 variables. From this array of correlation, hung on the phytochemical aspect of phytobiome studies, they isolated statistically significant relationships between phenolic compounds content and other variables. This culminated in the compensation matrix featured on the right of the slide. Notably, the evaluated metabolites showed no correlation with any mycorrhization metrics. However, the available zinc, phosphor, and sulfur, soluble phosphor and magnesium, and calcium saturation exhibited significant negative relationship with total flavonoid content, TFC. This suggests that great nutrition availability or soil fertility may correspond to the lower expression of phenolic compounds and vice versa. These nuances are crucial to consider, especially with the species cultivation and subsequent use of its leaf to the extract. These compounds are of interest. It implies that intensive fertilization may not be necessary to achieve desired total flavonoid content yields. Interestingly, in natural population, Gouda growing in nutrition-poor soils tends to exhibit increased flavonoid content levels. We are merely scratching the surface in understanding the intricate interactions within the heart of the Guadua phytobiome, with Guadua angustifolia emerging as a prime candidate to deepen our comprehension. These advancements are poised to not only enrich basic scientific knowledge, but also contribute to the well-being of ecosystems and human population. Thank you.
Thank you, Monica. No. Any question? So, if you have any question, this is Luciana email, and you can write to her. Yeah.
Don't ask me, unless it's about orchids.
I provide the material and I identify the Guadua species. That is because I am member of the team. So I don't know. Yeah.
Thank you all for participating in this